Thank you, Paul. So if you missed any of those details, I promise you the entire day is structured around all the exciting initiatives that Paul just mentioned. So if you're wondering, well, I want to know more about the immunotherapy initiative, or what exactly is the Answer Fund, trust me, you'll hear, if you look at the agenda, you'll hear a lot more about it. And I really wanted to emphasize that the reason we want to connect all patients, the reason we need to connect all patients, is because this disease is so heterogeneous. You probably know, at the cancer center, you meet someone else who's on a different therapy, who has different side effects. This disease is very heterogeneous. So it's one thing for you as a patient to understand your own data, and that is really, really important, to make sure that you understand the key numbers on your test results. But it's also important to see that data in the context of many other patients. And so when you have these subtypes that may only be 5 or 10% of patients, it takes a lot of patients to get there. It takes a lot of data, and it takes a lot of brilliant minds analyzing that data so that we can identify the different subtypes and get precise therapy to each of them. So this first panel will do that. We'll talk about data generation, a lot about the COMMPASS study, and also about the analytics that need to happen to drive it to the clinic. But before we do that, I will share a few slides with you on the topic. I think most of you are familiar with the MMRF COMMPASS study. It's the cornerstone of our precision medicine model. It's a $40 million effort, spanning at least 10 to 12 years. There's more than 1,100 patients enrolled from 76 centers in 4 countries. We've collected a lot of data so far, as you heard from Paul. It's now the largest genomics data set of any cancer. And for those of you who were paying attention last year, it was only the second largest. So in that year, we've amassed even more data and surpassed breast cancer. Just a little bit about the COMMPASS study. One of the things that makes it very special is it's a true snapshot of myeloma in these 4 countries at least. And I don't know if you can read the screens, but this is a chance to look at your iPad. Most patients with myeloma tend to be in their 60s and 70s, but we know it could span from teens to 90s. And indeed, in the COMMPASS study, the average age of a patient is 64. We had a patient, this is most recent, 27, enroll in mid-20s all the way to 93. Myeloma is more common in men. Indeed, we do have more males in the COMMPASS study. And one thing that we're really proud of is that we have representation across ethnicities. This is a disease that many of you know, that African Americans have twice the risk of other ethnicities. But if you look at clinical trials, quite often they're overrepresented by white, more affluent, more educated patients. It's really not representing the true snapshot of what myeloma is. So again, we're really proud that COMMPASS represents what patients truly look like who have myeloma. And then as the data has matured, and again, this is probably a good one for you to look at your iPad, we're learning things. We're learning things about the entire population. We've seen so far, for example, and it may sound intuitive, but this was a big debate in the field for a long time, that patients who start on 3 drugs do better than those on 2. Patients on 2 drugs progress faster. But what we'll be able to do over time is to say, okay, which patients on 2 drugs did perfectly fine? What kind of disease is, low risk enough that can be managed with 2 drugs, and you're not incurring additional toxicity, additional cost by giving that 3rd drug, versus which patients are still on 3 drugs and are still relapsing very, very quickly, who aren't responding very well. And that's a lot of what we've been able to look at so far in terms of identifying what high risk looks like. Similarly, and I heard a little bit of a reference to this in the film, we've seen that patients in general, again, we need to break it down by subtypes, but in general, patients who go right to stem cell transplant, again, have a longer, what's called progression-free survival, time before the disease comes back, than patients who don't. But again, these kinds of analyses need to happen where who are the patients who had a transplant but never responded, or who relapsed very quickly, versus who are the patients who didn't have a transplant and are out, with a complete response years later. That's what COMMPASS will allow us to do, really drill down to subsegments of patients to be able to deliver on that vision, telling every patient what treatment he or she needs along the course of his or her journey. But really, we started COMMPASS to identify these subtypes. And I love this story. Daniel has presented this slide multiple times, where you have two patients who look pretty much identical. They came from the same center, they're the same age, same ethnicity, they started on the same treatment. If you use today's tools in terms of assessing risk, they look pretty similar. But what's different? Their outcome. You have one who is still in a complete response and one who relapsed very quickly. But, again, with today's technology, you would ask why. With COMMPASS and the kind of sequencing that we're doing, we can see that deletion 17p, you've probably all heard of that in terms of high risk. What COMMPASS has shown us is that with better technology, we can differentiate between these deletion of 17p patients. Jonathan Keats, who many of you have heard from at these past summits, uses a great analogy. P53, related to deletion 17p, is a tumor suppressor gene. It's like brakes on a car. You cut off one set of brakes, the car will still stop. You cut off both, the car won't stop. So what we've been able to differentiate in COMMPASS is those patients who have one copy still intact versus those who have none. With current technology, you can't do that. So again, we're learning things that wouldn't have been possible without using this deep sequencing through COMMPASS. So with that, I'm thrilled to turn it over to our panel. We have Bob Tedeschi here. He's a senior writer at STAT and a longtime healthcare journalist. Joining him are a variety of perspectives. We have, you guys can start coming to the stage. We have Colin Hill, who's chairman, CEO, and co-founder of GNS Healthcare. He's been a speaker, and some of his colleagues have been here before. GNS is an important partner in terms of driving the COMMPASS analytics, applying machine learning and AI to mine the COMMPASS data. Robert Loberg, who's our partner at Amgen. He's head of clinical biomarkers there. Many of you know Amgen makes Kyprolis or carfilzomib, and has other very exciting agents in the pipeline for myeloma, and he has been an incredible champion for COMMPASS. And then last but not least, Sagar Lonial, who's professor and chair of hematology as well as chief medical officer at the Winship Cancer Center at Emory University. He's also principal investigator for COMMPASS and co-chair of our clinical network, the MMRC. At the end of the session, Chris Williams, who's our VP of Business Development, will share some details about the exciting new initiative, the MMRF Answer Fund. Chris joined us earlier this year, 2 months, I think you said. He spent a long time in biotech diagnostics. He was at Millennium when Velcade was approved, and he's been extremely passionate about bringing his experience to bear for the MMRF. Now I'll turn it over to you, Bob. Thank you.
Thanks so much, Anne. That was a great overview, by the way. The COMMPASS study is clearly one of the biggest innovations to come out of the medical foundation space, period, and it's also come at a pivotal time in history given the exponential increase in computing power, which itself has given rise to a swarm of data collection and all the talk around big data and machine learning and what we could do with this data, how to mine that information for the benefit of patients, right? Those are all big and sometimes tough discussions to follow, unless you've got panelists like Sagar and Robert and Colin who are exceptional when it comes to translating these sorts of topics for people who don't spend their days in data analytics and bioinformatics. So thanks again to the three of you for joining us this morning. I want to jump in with since it's early and the coffee hasn't fully kicked in, sort of a 30,000-foot question so we can sort of set the context here when it comes to big data and drug discovery. And I'd like to open it first briefly to Colin and then ask Robert and Sagar to join as well, and ask very broadly how big data is being used to drive drug discovery, even outside multiple myeloma, and how quickly that's been changing over the course of the last several years or so. And then again, I'd like Sagar and Robert to follow up. So Colin, please.
Sure. So I think that one of the big changes that's occurred in the last decade is the fact that we can now measure tissue at a genomic level. So instead of just measuring activity of a gene or protein one at a time, we now have a chance to get really a panoramic view of say, cancer tissue versus normal, drug treated versus not. And this is enabling a different kind of drug discovery than in the past. There's now the ability to use this data to reverse engineer even computer models of how all of the genes and proteins connect to each other and drive clinical outcomes. This is a big difference from, say, a decade ago or 20 years ago, when we certainly didn't have the data and we didn't have the computing power. We happened to live in the shadow of many decades of Moore's Law that give us ultra-fast, ultra-powerful supercomputers on the cloud. And when you combine that with tools from machine learning and artificial intelligence, and there's many different types of tools there, there's now the ability to make computer models, not just even analyze data and find patterns and correlations, but now even start to use the data as fuel to make computer models of the disease itself, of disease progression on a mechanistic molecular level, such that we can now, instead of just hunting for our favorite genes, we can now have the computers simulate interventions, knockdowns, gene by gene, protein by protein, and in a hypothesis-free fashion, discover new drug targets, new pathways for intervention, and even discover markers of response. And that connects back to the key questions. Should I do a stem cell transplant? Yes, no. Should I take this drug versus this drug? Yes, no. Fortunately, there's a lot of options these days in terms of drug treatments, and it's now a question of, well, which drug for me? What's going to have the greatest impact on me as an individual versus the hypothetical average patient? So that's, I think it's the personalization of drug discovery is one, and two, the fact we can now start to tackle the full scale and complexity of biology in a way we just couldn't have touched 10 years ago.
And how much of that is promise right now versus reality? And any three of you can chime in on this as well, because it sounds like a terrific promise to be able to get to that level of precision and prescription. But how much is that actually happening?
Yeah, I'll start on that.
Okay.
It's so I'd say a decade ago it was still a hope and a dream, right? No one really had the data. The computing power wasn't there. The tools just weren't there to really do this. I would say it's real now. I'd say there's a number of discoveries that have come from even databases like COMMPASS, but we're only scratching the surface, right? I mean, I am convinced that we're only touching maybe 5% of what is there to be discovered, both from this data and from a second version of this, especially when you start to also combine that with other real-world data assets. Think about all the electronic health record data that's out there in this country that has not been linked together, has not been linked to molecular patient registries like COMMPASS that offer a whole new basis for discovery. So I would say we're beyond the hype and hope phase, and the rubber is starting to meet the road, but I think we're still at the beginning, and we're starting to see the cycles go faster and faster. The cycles of data collection, of profiling, of computer modeling, of discoveries, of validation, and I think the next 3 to 5 years are going to be incredible for this field specifically.
Great, thank you. And Sagar, let's dial this into the COMMPASS, where the intersection is between this more theoretical discussion and the practical of COMMPASS. And then I want to ask about industry.
Yeah, I was going to contrast a little bit. I mean, that was amazing science, amazing computing technology. I'm a simple country doctor. That's what I tell people all the time. I have a PC at my desktop, and I'll give you a sort of a real-world example. We had a scientist who wasn't really a cancer biologist. He was just a regular basic scientist at our university who had a paper on a gene called MAX. And he was really interested in MAX because MAX worked with MYC, and MYC is really important in myeloma. He sent his paper in, and the reviewers sent it back saying, this is really neat, but what's the clinical implications of mutations in MAX? He had no idea. He happened to be at another meeting with one of our myeloma scientists who said, let's look in COMMPASS and see what MAX does in COMMPASS. Well, it turns out MAX mutations predict for better survival. And so that led to a whole nother series of experiments that have now put MAX on the table as a target in myeloma. Because if mutations or changes in MAX, which is a gene none of us really think about in myeloma, he didn't even think about myeloma. He's a basic scientist thinking about mechanisms of disease resistance and survival. All of a sudden now MAX is on the table as a target in myeloma. And this guy is a pharmacologist who's thinking about what's the drug look like that targets MAX. That all came from a completely serendipitous interaction at a meeting of somebody who had access to COMMPASS that put that science data in the context of patients. And that's, I mean, that's a simple story. There are many others.
Great. And Robert, how about from an industry standpoint, to what extent are you able to tap into COMMPASS, or to what extent and at what pace have you been tapping into, and how's that changing?
So data sets like COMMPASS are extremely important for a variety of reasons. One, we're learning biology of disease, in this case myeloma, at such a deeper level than we ever understood before, and it's allowing us to not only explore potentially new targets, but also understand the targets that we're actually already pursuing in the clinic with some of the novel therapies that are coming through, and how those markers, those biomarkers change through the progression of the disease, because that becomes very important as you're thinking about where you can actually help patients with some of these novel therapeutics. Not all targets are the same at all phases of a disease or disease progression. So using data from studies like COMMPASS really help us understand, again, the biology in such a different way than we've been able to in the past, that it's really transforming the way industry is really being able to pursue and generate that conviction around a target in a disease setting to bring it forward and help patients.
Interesting. Can you point to any examples where this is where you've kind of walked through the way Sagar did, a time or a place where you've been able to tap into COMMPASS and said, you know what, this may not have been possible had it not been for COMMPASS in this moment? Or can you speak to that publicly? I know some of this can.
No, it's fine. So perhaps a little bit more generically, I mean, there are a number of targets that we and a variety of other industry folks are pursuing. And using COMMPASS, you really start to, again, understand where it's possible to bring these molecules forward that are trying to interdict these novel targets, to really understand how to get these medicines to patients as quickly as possible. One of the challenges going back, pre-10 years ago, is the concept of you kind of had to try it in the clinic without a lot of information with respect to a patient or a patient's genetics or expression level of the target you're trying to interdict, and just see if the medicine works based on some understanding that you have from some models that you run in a laboratory. Now we're actually able to look in data sets like COMMPASS and ask hypothesis-driven questions around some of these novel targets as to where we should take them within the setting of myeloma, again, to really prove whether or not they are effective and get them into patients' hands as quickly as possible.
And if you think about, sort of to take that even one step further, so one of the most exciting, I think, immunotherapy targets in myeloma, and I know this will come up in the immunotherapy session, is BCMA. BCMA, and you'll hear a lot about it later on. CAR T cells are going after BCMA, antibodies are going after BCMA, BiTEs and bispecifics are all going after BCMA. It's probably the best immune therapy target in myeloma. Well, a company came to us and said, do we know anything about expression of BCMA between newly diagnosed and relapsed myeloma? And previous to this, there had been a small series of patients, everybody's got their own set of 20 or 30 patients. We looked in COMMPASS. We looked in COMMPASS at expression of BCMA by RNA at the diagnosis, and then in the 200 or 300 relapse samples we have, and it's pretty much the same. So that's a useful piece of information, because it tells a company you can go at this target at least in the early relapse and in the newly diagnosed setting. It doesn't speak to late relapse when the expression may fall off, but that's a useful piece of information for a company when they're designing a trial, and for us when we're thinking about who should go on that trial.
What makes it, and Anne pointed to this a bit in her presentation, but I'm curious from a layperson standpoint, what makes the COMMPASS data so powerful? I have a feeling that there are a few different elements, but if you could please elucidate a little bit about why that data is such an important data set for researchers across the community, and then this could be anybody.
I mean, I think it is the largest data set of molecularly characterized patients in the world with myeloma. Nobody else has this depth of molecular information with follow-up. So it's great to say in a snapshot that if you had 1,000 patients, X% of them had a mutation in this gene, and Y% of them had a mutation in that gene. But what COMMPASS allows you to do is to say, yes, they had this mutation, and when they got treatment A, this was their outcome. So that progression-free survival, overall survival, longitudinal follow-up is what really differentiates COMMPASS from many of the NCI-sponsored TCGA approaches, where it's just a brute force, here's 1,000 patients, tell me about the genetics. That's interesting, it's good, but it doesn't give you the follow-up and clinical implications of that genomics, and that's what I think COMMPASS really brings to bear.
And maybe to add on to that, completely agree with all that, it's also, from a computational point of view, it's the size of it, the number of patients, the amount of diversity, including diversity of the interventions, that makes it so rich and so powerful. The other big aspect is that it's multi-layered, right? It's not just one type of molecular profiling view into patients. It's multiple layers. It's gene expression, it's sequence variation, and that makes it really rich and powerful in terms of being able to turn that data into the discovery of mechanisms, the actual mechanisms driving progression.
Mm-hmm. Yeah, maybe just last 2 points. A couple concepts that weren't mentioned is the longitudinal sampling. So every time a patient progresses, right, their bone marrow is recollected and resequenced, and being able to follow a patient's disease course following treatment as they progress through their disease at a genomic level is something that I think is incredibly powerful about the COMMPASS trial itself. But then also the foresight to collect and biobank, I think Paul mentioned earlier, the biobanking efforts to actually bank some of these samples so that we can go back to those samples and test some of these hypotheses within patients or patient segments that have specific characteristics that we want to understand a new hypothesis in. One of the problems that we face within the scientific community, not just industry, is being able to have access to these relevant samples from patients that are very specific in either the genomic profile or what treatment they were on or what stage of the disease they're at a sufficient scale where you can actually formally test a hypothesis, a new hypothesis, to be able to make a conclusion and kind of take a step forward. So to me, that's a very powerful piece that COMMPASS brings.
And as an example on that, so there were platforms that were used for the genomic and the genetic sequencing. Three different platforms were used to speak to that richness of data that's available for every given patient. We now, through collaborations with the foundation and with the MMRC, are going to be doing what's called epigenetic profiling. That was not in the original plan, but because of the biobanking that was done on over 800 of those patients, we can now do the largest series of epigenetic profiling, which is another way to assess whether a gene is important or unimportant based on whether it's turned on or turned off in a completely different pathway that has never been done in this many patients before. And the first 95 patients just arrived at our place last week. So that whole process is going to start now in real time to add to the data complexity.
And in a few minutes, I'll ask you to further peek around the corner of where we're going with this data set. But I want to backstep just a little bit and ask a point related to the longitudinal element of COMMPASS. I've spoken with a bunch of different researchers in other cancer areas who are completely frustrated at the fact that they can't get longitudinal data, and it's so critical for their research efforts because when somebody relapses, when somebody progresses, it's a completely different cancer, and that's the moment a lot of times, or their final relapse, that's the moment where they could learn the most from patients. And it seems like in the multiple myeloma community, that is if not unique, it's very unusual to have that data set. I'm curious whether or not what you learn from that longitudinal view might extend to other cancer types as you keep looking and checking and watching this cancer mutate. Is any of that extensible to other tumor types, or is it just within the realm of multiple myeloma only?
I mean, I think it is. There is a large amount of that information that can be generalizable to cancer in general, and some of that gets back to the point you made up. It's not just diversity of patients that gives COMMPASS its power. It's some level of diversity of treatments. So everybody didn't get the exact same thing in the COMMPASS trial. And I can tell you, having been there at those early meetings where we were designing this was an hour-and-a-half long discussion. And discussion is the nice way to put it. It was a knockdown, drag-out fight between about 4 of us who said, I said everybody should get the same thing. I was wrong. That's the only time you'll hear me say that. And then many others in the room who said, why don't we let people pick what they want to get, and this way we will have that richness of diversity in terms of treatment. So what we now have in COMMPASS is when patients relapse, if they relapse, we have their sequence at relapse, and we can see what impact the treatment had on the changes in mutations. And the treatment may have had some impact, it may have had no impact, it may be the original deletion of 17p that had the greatest impact, but we couldn't ask those questions if we didn't have this kind of longitudinal follow-up. And that's generalizable.
That's great.
Yeah, I think it's both the framework of that type of longitudinal data with diverse treatments, but I also think on a deeper level, there's a lot of common mechanisms between different cancers. We know that the cell signaling that initiates the replication of cells is conserved amongst many cancers, many tissue types. We know that mechanisms of apoptosis or programmed cell death are conserved. And so if you not so much look at this as let's try to analyze data, this cancer and these various other cancers to find patterns, but you think of it more as the data is fuel to reverse engineer an actual mechanistic knowledge of what's driving these cancers. And then you really do have a pan-cancer way of looking at the world, where when we discover certain connections between genes or between treatments and genetic background characteristics of patients and outcomes, you really can start to translate across many of these cancers. So not just the paradigm of a longitudinal data bank, but also the actual discoveries themselves. And I don't think this has been exploited nearly as much as we can, because people tend to work often in silos, and I think there's a chance to start to break that. And I think what Kathy's doing even at Harvard is looking more broadly than multiple myeloma.
It'd be pretty remarkable if it started in multiple myeloma and you'd had some broader discoveries across the spectrum. Robert, anything to add before I?
Maybe just quickly, I mean, I don't think we really fully understand how all of these genetics around cancer as a whole relate when you look at very specific subtypes of cancer. The MAX example is a very good one in that was something that was discovered that didn't even have a myeloma story to it. But having something like COMMPASS, where you could go in and ask the question and very quickly come up with an answer that maybe this is something that we could pursue in myeloma that would actually have quite an impact for patients, is very powerful. So I think what we'll find over the coming years, and this kind of goes back to an earlier question of where are we, I think we're at the doorstep of really understanding this large-scale big data, genomics data as it relates to cancers. We now have the technologies, we now have the platforms to collect the data, we're building all of the tools to analyze the data in a very comprehensive way that we've never been able to do before. And now we're going to be able to start asking the questions, how do they translate across diseases, not just within myeloma? But you have to build data sets within an indication first, right? Or indications, plural, to be able to ask those questions. And I think we're kind of just in that era where we're going to be having those types of abilities.
Let's peek further around the corner, and I'm curious to know where COMMPASS goes next, or projects like COMMPASS go next when it comes to harnessing the discoveries around machine learning and AI that are happening separate from the medical field, and how fast that's growing. And then, as the COMMPASS data set grows and gets more sophisticated, what are the possibilities that we're not seeing quite yet that could be brought to bear on multiple myeloma research in particular, or cancer research generally? Because we know that machine learning has just got a brute force, a huge army of research going behind it, separate from this. So Colin, what do you think about where that's headed?
Sure, and yesterday we discussed some of this, and it goes to the fact that each sort of layer of data is a different snapshot into the disease. And so when you can analyze DNA sequence variation, you're seeing an important view, but you're not seeing a complete view, right? So gene expression adds another very important view of what's going on in the disease. Epigenetics that you were describing. But take even immune the measurement of the immune status of patients, these are going to, it's going to add a level of richness that I think it's hard to anticipate how powerful it's going to be, because it's adding on top of what's already a very rich database. And so if you take a hypothesis-free view of the world, which machine learning folks and AI folks do, then as we both increase the number of patients, as we take the N from 1,000 to hopefully 5,000, 10,000 soon, as we add in these other layers that start to give us a different view of what's happening in the disease, I believe that it's going to accelerate the pace of discovery by a lot, because the machines are getting faster, meaning the actual computing power is getting faster and cheaper at a rate that very few things double in power every year, every 18 months. And so I do think that where COMMPASS is going to be disruptive at a pace that's faster than what human experts can do. I'm sort of the, let's say our company sort of represents the view of what can the data tell us above and beyond what a human expert can see from these components. Because ultimately, I think the real battle, the real war here is the complexity of these systems. You have thousands of genes interacting in complex ways with other genes and gene products, with the potential for many billions of pieces of variation patient to patient. This is beyond the smartest human in terms of really being able to fully understand this. Now, it's not to say that human experts have a huge role to play, and it's really the interplay between the machine learning and the human experts that I think is going to win the day, and probably faster than we think.
Sagar, what are your thoughts on that, especially with respect to the human element interjected into artificial intelligence and machine learning?
Yeah, I mean, I think the ability to analyze large pieces of data quickly and efficiently is something that I don't think any of us, even the smart of us, can't do, certainly at the speed that can be done by machines. But all of that has to be put into context. And many of the diagrams that say these genes are connected to each other come from our experience and our laboratory work, not from a computer telling us that these genes necessarily interact together. So I think the interface between that huge computational power and science, which is hypothesis-based science, is really the key to moving this piece forward. And I don't think either of the two pieces could do it alone. I think it really is the partnership between the two that'll be the success.
Yeah, I completely agree. You know, it's said that humans can track somewhere between 8 to 15 concepts as they're trying to solve a problem. And when you think about the massive amount of data that's generated out of some of these sequencing and other types of assays, I mean, it's literally millions of data points that are somehow connecting together to explain the biology of the disease. And so being able to use machine learning, AI type of principles to really get down to some very tractable hypotheses that then you can formally test in a laboratory setting to help us advance our understanding of the biology is really where the power comes from.
With every sort of technological leap, you've got some naysayers, of course, and you've got some people resisting for cultural reasons, whatever it might be within their profession. And I'm sure that medicine is no different. I know that there are some people who resist the lure of or the hype around machine learning and AI, and yet those on the forefront of it can see so clearly how this is going to dramatically change the pace of innovation. So how do you communicate to the frontline physicians who have access to those patients you need the data from, how that technology and machine learning are something to embrace and run toward rather than resist? How do you make that kind of framework shift? Anybody?
Well, I think it's going to take I think everybody is willing to say we don't have the answer right now. So I don't think there's a hesitancy to say this may generate some new ideas and new approaches that we can take that will be useful on a day-to-day basis. I don't think that for the frontline clinician that's seeing a newly diagnosed myeloma patient, that AI right now is going to impact how they're going to treat that patient. But I think, I don't know anybody that's not open to the idea that if you can give us ways to think about pathways that we can't begin to comprehend with a data set this large, we're open to suggestions. I don't think any of us think we have the answer.
Yeah, I would say that translation and collaboration and humility are also key to making this work, because it's to be able to translate between, deep experts in multiple myeloma on the clinical and the biological front with the deep experts in computer science and physics, driving the machine learning. And that's really key, because by themselves it's just not gonna get as far, and that does mean collaboration. I think one of the things that's been done at MMRF is to really force strange bedfellows. I mean, it forces collaboration amongst groups that otherwise may not work together, because you're going after a problem that's so big. The only way you can solve it is for it to be collaborative across many clinicians and researchers, but also different types of researchers who approach it very differently. And that's, I think, also where the humility comes in. It's, there's room for lots of ego in any profession, but I do think when you're working across these lines of disciplines, it's important that people go into it with being very clear of what they don't know, and being open to just learning from each other.
Robert, anything to add?
I guess lastly, I mean, it's just a constant education of the value proposition that will come.
And along those lines, this is a kind of a question dovetails a little bit. What incentives or deal structures are needed to combine COMMPASS with other data sets, possibly to increase the power of the data, and how feasible is that right now, given how competitive the research field still is? It seemingly will always remain.
So I don't know that you need incentives. I mean, I'll tell you, the incentive for me is seeing a patient do better in the long term. The patient I treat today does better than the patient I treated 5 years ago. I don't think we in the research community need incentives. I think we're doing that. There are other data sets that don't have exactly the same level of complexity or depth of data, but do have comprehensive sequencing data that are beginning to emerge now, that can be combined with COMMPASS to really increase the power of observations. And as you said, when you do a study of 500 patients that have been sequenced, most large data people say that's a good start, right? I mean, the ability to take 1,000 and make it 1,500, 1,800, 2,000 just increases the power and the ability to ask really big questions that you can't ask with 100, 200, or even 500 patients.
And some of these really big questions that require really big data sets now push you into the real world, because there's only so much money and time for doing deep data collection like COMMPASS, and so you want to complement that with access to, and the marriage to, electronic health records and other types of data. But there are impediments to that, right? This is where you do get into regulatory issues around patient privacy and HIPAA at a federal level that I think start to be not in the best interest of patients and researchers when it becomes super difficult to get access to data that when you connect it up to other types of data will drive real discoveries. And the question is, who is this protecting? And I think we've crossed the line of where that's maybe going further than it needs to be. Patient privacy is super important, but let's not do it to the extent that it impedes the pace of discovery.
Robert, how about you?
Yeah, no, I guess lastly, I'm very excited in the position that I'm in within industry. I see a lot of collaboration amongst industry as well as foundations and academicians that are really trying to answer this question. I don't proclaim to have an answer for you today on how to make it easy, but the momentum is clearly there. People want to pool data, pool resources to be able to answer questions in a way that we haven't been able to answer them before. So, I am very excited about where we are within the scientific community. There are efforts not only through MMRF, but through some of the other big oncology conference and organizations like ASCO that are trying to do very similar things, open up data sets to be able to be combined and be able to answer these tough large questions. So
Because there are silos, I mean, I imagine maybe not as good as COMMPASS, but there are silos of data out there
Sure.
that could conceivably help you build 1,000, 3,000, any number of more patients into your data set, right? And the question is whether or not those discussions are becoming easier, and if so, at what pace, or is it still kind of a slog to reach out to a counterpart at another foundation and say, hey, let's talk, or let's get these two silos to connect somehow?
Yeah, I think it's getting easier. I mean, I think the combination of data, usually from multiple large trials, is the way that it's going to get done. So you've got the COMMPASS trial, which is fairly sizable in and of itself. There are other trials that have been done with homogeneous treatment approaches in the context of a clinical trial, where they do have sequencing data as well. Those are now being put forth to be combined with COMMPASS to get a larger data set to answer some of these questions. So I don't think it's harder to do. I think people are very open to this idea, especially because COMMPASS bears a bulk of the data. So everybody wants to combine with COMMASS because it adds power to their study, right?
Nice position to be in.
Yeah,
Exactly.
right.
Okay, so I'm going to open it up to questions of the audience after this one, the last question. I'm curious, what are the most important, in each of your opinions, questions that researchers or clinicians and industry have that COMMPASS could answer going forward? Like, what haven't you pointed it toward, or about to point it toward, that you think could yield something in the next 2, 3, 4 years for patients who are on their second or third line of treatment?
So I guess from my perspective, it's the longitudinal sampling that is really changing the way and our ability to ask questions about various targets that we're pursuing and how they relate to the disease course. So, I mean, that is a very, very powerful tool that's really helping drive, I think, how we look at myeloma.
Any specific questions that you can talk about within that realm, or is it all on a need-to-know basis?
I don't know about need to know, but I don't know if I can give it a specific.
Okay, I won't push you. Colin, how about you?
I think treatment effectiveness in a patient-centered way, and this even goes right back to the efforts that GNS is doing in collaboration with MMRF. So far, it's been very focused on the discovery of new therapeutics, the discovery of markers of progression, so high risk versus not high risk. I would love to see over the next couple of years that the computer models start to really get turned into in a way that can be used directly by patients as really clinical decision support, where you can, in this virtual model, press the button based on your own specific data of what would be the likely outcome for me under this treatment versus that treatment versus this combination. And this is a very big and lofty goal, but I don't think it's that far away from being useful. It's not going to be perfect, but it's going to be useful. So that's what I think COMMPASS can support that now, and I think it's going to do it that much better going forward.
Right, a little bit what Dick Parsons was talking about in the video, right? A little bit, but much.
That's right.
Probably
It's exactly what he was pointing to, which is the big question for every patient, right, is what happens if I don't take that path, or if I go this way, or if I go that way. What are the likely outcomes, not the statistics? I mean, this is what I think Ann was pointing out at the lead-in to this panel. We see these waterfall plots that are giving important answers across the board, but what about for the individual, right? Because that's the average, and what is going to be the impact of this intervention on this given patient? Because stem cell transplant is not going to have the same impact on everybody. It's not always the right answer, similar with the various therapeutics we have available.
And making it smart enough so that that's not misleading information, right? It's not that person
correct.
is like me in 12 different ways, and 12 is not nearly enough.
Right.
That's right.
To make this an intelligent call. Okay.
Right.
So-
Yeah, I mean, I think so. There are two extremes that I think we can already begin to tease out right now, and that is the NCI definition of the exceptional responder. people that do remarkably well. We've all seen them. We've all had them. I had 2 in my clinic on Tuesday that are 10, 12, 14 years out from their transplant and have done phenomenally well. And I would argue that many of those patients are probably cured. What do they look like genetically? We'll know that in a few years as we get more data on longitudinal follow-up. And the flip side of that is the exceptional non-responders. So the people that relapse really, really early despite getting highly effective therapy. You can argue we know who high-risk people are. We have FISH testing, we have cytogenetics. But FISH testing and cytogenetics is technology circa 1990s, 2000. We know that everybody's going to be getting genetic profiling with sequencing, RNA-seq, and we need a data set that establishes what is the high-risk signature with RNA-seq, and that's what COMMPASS does. There was a paper published recently from Mount Sinai that probably has identified some of those things. This will be the benchmark against which other trials are compared and will be the sort of curve against which we show, did this intervention make a difference or not? Because this is the largest data set of those short responders we have.
That's a fascinating area of study. Okay, some questions from the audience. We ran right by this. How does COMMPASS collect patient data?
How? You mean the actual mechanics of how it
Yeah, I'm
happens?
guessing so. Like, how does that work?
So-
Like, who makes the ask, and then what does that involve from a patient standpoint?
So COMMPASS is technically a clinical trial. So clinical data is collected in the same way it is on any clinical trial, which means a patient comes into clinic, say, to see me. There's a research coordinator or a nurse that goes and visits the patient. They get the labs, they get the story in terms of side effects and management. That gets put into what's called a case report form, a standard form. That case report form gets uploaded to a database, and that database then exists for each patient over time. And part of what makes clinical trials so challenging sometimes is the fact that what is put into the database may not always be correct. So somebody has to go back around and audit that data to make sure that what you're looking at is actually good data. And that's the part that takes time and takes, it's expensive for companies when they run clinical trials because, I may enter a 4 instead of a 7, and it shouldn't be a 4, it should be a 7. I don't actually enter the data, so that should make you take some comfort. But the people that do it do a lot of this, and so it's a process that has internal validation to make sure the data is robust.
These are great questions, by the way. So much of medical research and the movement forward is based on genomic profiling and sequencing, right? Yet a small fraction of patients actually have this done for a multitude of reasons. Chief among them, nobody wants to pay for it. So how do we get to a place where genomic sequencing is the standard of care, is the norm for 90% of patients instead of, what are we at, 3, 2, 1? Whoever wants to whack at that one.
I think as it gets cheaper and more robust, it will be used more broadly. But to be very practical, from the point of view of health insurance companies and the payment for such testing, when it becomes cost effective from their point of view, you're going to see it be very widely covered. And they look at it from the standpoint of there's a wide variation in the cost of treatment, drugs and drug combos going up to $250,000 a year. If some of those expensive, not effective treatments can be avoided by doing broader diagnostics, then you're going to see the big health insurance companies start to, I think, adopt it a lot faster.
You can make the argument that that's now, right?
Yeah.
I mean, they could be using this. This is very actionable information right now to make financially, intelligent decisions. But that's not quite. We're not quite there yet. Right?
And I think this goes to the whole debate of drug prices and are drugs too expensive. I think it's often the wrong question. Like, let's first make sure people are getting the right treatment that are leading to the right outcomes, and because that's really the first thing to get right, versus what should a drug cost or not cost. It's let's stop wasting huge amounts of money where it's no benefit to the patient, it's no benefit to anybody.
Right. So I mean, I think I think you have to know what's the comparator. So if you're talking about sequencing not being the standard of care, which I would agree currently it is not, but I think it will be very soon. The current standard of care is FISH testing with cytogenetics, which FISH testing is a way to look at the bone marrow. Many of you know this acronym. It's $500 a probe. We do roughly 8 to 10 probes per patient. So that's $4,000 to $5,000 for FISH testing at the time of diagnosis. Keith, how much is your panel? $2,000 for his targeted sequencing panel. So already there's a cost benefit. What we need is the validation that you get the same information or better. And we know that, but we need the data to really feel comfortable in a large trial to say that the targeted sequencing, which by the way is cheaper than FISH testing, should replace FISH testing.
That separation between what we know
Yeah.
and what we have data on is so troubling. Just compressing that is, it seems to be the key to so much. So what skills do newly educated computer scientists need to master to assist more in the big data effort to move the fight forward faster and more effectively? I'm gonna guess that Colin might be able to speak to this most effectively.
So from the point of view of computer scientists and quantitative types, what do they need to
If you had a
supplement?
child you wanted to point in the direction of drug discovery to speed things up, where do you put them?
Well, it would be great if they learned some biology and genetics, if they're coming out of computer science or physics, and then you can kind of say vice versa. If they're coming from molecular biology and such, it would be great if they learned to program, and they learned statistics. But I'm less into the formal degrees that are kind of the flavor of the month of getting degrees in bioinformatics or this type of thing. I think it's more just experience. I think it's to have people who are well trained, whether it's in molecular biology or computer science and physics, and get them actually solving these problems. I think the folks in this are at the forefront of the field. They really came into it by just doing it and learning and applying the tools of their trade, and eventually they put in the 10,000 hours and became fluent enough to talk to experts in various diseases. Because I think that's the key. It's not that the computer scientists are going to become the best molecular biologists or the best myeloma doctors, but they need to learn enough to be able to collaborate and interact.
Mm-hmm. Last question. If researchers are going to combine data sets with COMMPASS, can those efforts go back to the COMMPASS portal so those combinations are in the public domain as well? I mean, it's probably a case-by-case basis, but since you've got leverage because you've got the gem, can you use that as leverage to make it public?
Yeah, I mean, I think that would be the goal. I think it was a bold step to say everything in COMMPASS is going to be publicly available. That was sort of a paradigm-changing approach because typically those large data sets have been released once the data is published. That's a requirement by many journals, but they've not been released as early as COMMPASS released the data. And I think that kind of leverage is something that can be brought to bear to those large collaborative efforts.
Well, I'd like to thank these panelists. They're tremendous, and thank you for your questions as well. We look forward to having more discussions offline.
Great, thank you.
Thanks.
Thank you.